my name is Sebastian Wieneke. I'm basically started my PhD in 2012 at the University of Cologne together with Uwe. And so it was like the early start of sun-induced fluorescence research. And my main focus is on proximal remote sensing. So we started with the first versions of measurement systems, which were able to measure continuous sun-induced fluorescence. And then after my PhD, I went to the University of Antwerp on a Marie Curie to go more like into manipulation experiments and to see like, okay, how much can we stress a plant to actually get nice signals in fluorescence. And since 2020, I'm a postdoc at the University of Leipzig in the Remote Sensing Center for Earth System Research with Miguel Maestre. They are basically like setting up new measurement systems in different ecosystems, or like mainly in forest actually. And there we compare different tree species with each other and to see like how structural influences actually make it our life a little bit more difficult in interpreting sun-induced fluorescence. And this is basically what I also would like to talk today about, which is the challenges, but also opportunities and what works, what did not work with interpreting sun-induced fluorescence. I would like to talk today about interpreting sun-induced fluorescence under stress conditions, and I will guide you through basically my experience with the signal through my PhD and my postdoc years, and what I think, what I learned and what we learned in the community, and what are the current challenges. First, the question, why do we care at all about fluorescence? And what you see here is now actually reflectance data from Sentinel-2. That is the lines. The blue line is the K NDVI, and the red line is the inverted red edge chlorophyll index. And the dots, they show GPP data from an eddy covariance site in the Hainich National Forest. And then for the year 2017, which was a relatively wet year, and then 2018, which you probably know was a very intense drought stress year. And what you can actually nicely see is that when we look at reflectance data, we get a quite good idea about the phenology of plants and also about basically then also GPP. But when we get into stress events, and this is what you nicely see here with the onset of the stress, that the reflectance indices have a lag effect. They react much later. And basically the reason for this is we observe here parameter, which is just the reflectance, and it's not directly physically linked with the photosynthetic process. And that's the big advantage of sun-induced fluorescence, because as many of you maybe know, sun-induced fluorescence is emitted directly from the photosystem. So if we zoom into a leaf and then into the chloroplast, and then into the photosystem, so like into electron transport chain, you can see that fluorescence is actually emitted from photosystem 2 and photosystem 1. And this is what you see on the right side is the emission spectra of fluorescence, which is emitted between 650 and 800 nanometer. It's a relatively small signal, so we have to do some tricks on the retrieval to actually get the signal. I will not talk about this today, but we can retrieve fluorescence at 687 nanometers, so close to the red fluorescence peak, just first peak in the red, and to 760 nanometers, so close to the far red peak. And what is interesting about these two peaks is that while the second peak in fluorescence in the far red gets a signal from the photosystem 1 and 2, the first peak, the red fluorescence, the main contribution comes from photosystem 2. And that makes the red fluorescence very interesting because we have for photosystem 2 a closer link to the non-photochemical quenching process, which I will also explain later in my talk, and which is basically where the downregulation of photosynthesis happens. So the idea is that the red fluorescence, due to this stronger connection to the non-photochemical quenching, should actually give us also a more clearer signal about stress events and gross primary production under stress. First, I would like to show some results of my PhD time, and this is now on the far red because far red fluorescence is much easier to retrieve, and this is why also many studies actually focus on the far red at 760 nanometer. And what we know from fluorescence is that we actually get a quite nice relationship with GPP. This relationship is actually not linear, because we quite often observe that under very high GPP values, sun-induced fluorescence values, GPP is actually stagnating, and then we have like an increase in fluorescence. It's not very observable here, but sometimes we see that very high GPP values are not very well captured by fluorescence. But here we see a relatively nice correlation between GPP and fluorescence values. So each dot is a 30 minutes measurement from an eddy program station. So this is why the spread is quite large. But what we also observe is that under stress conditions, that's the red points, where the blue points are unstressed. Under stress conditions, this relationship is weakened drastically. And another point is that the relationship between GPP and fluorescence is also strongly driven by APER. So if we correlate fluorescence with APER, we usually get very strong correlations, because both depend on light. And this quite often defines this very strong relationship. And you can also see when we have under stress conditions, the photosynthesis is not light limited, and then this relationship also between fluorescence and APER is breaking down. What can we do to get closer to actually like the physiology, like what is driven by changes in the physiology and not so much by how much light is actually absorbed by the plant? We can look at the so-called fluorescence yield, and this is what we then also did in our experiment in 2015. We compared then the fluorescence yield with the light use efficiencies values. So we normalized by APER, and then see that this relationship becomes a little bit more tricky to interpret. We can find a nice relationship over the stronger stress gradient, but if you only look into the non-stressed or in the stress part, it gets difficult to interpret. And this is not only like with these results. If we compare it to other experiments, here from Miao, for example, we see different kind of relationships. Sometimes we see an increase with light use efficiency in SIF, sometimes a decrease. Here another example from Yang et al. In 2015. It's like strongly depends on how strong the gradient is we observe. Sometimes we get better results, sometimes not. But it's always quite noisy, or variances are quite high, and it's tricky to interpret. So the question is, why is that? Because as we learned, fluorescence should actually be like much closer related to photosynthesis. So one of the reasons is that the relationship between the efficiency values of photosynthesis and so the photosynthetic yields and the fluorescence yields, or the quantum use efficiency of fluorescence, they are not linearly related. So under what we know is that under low light conditions, and this is what we experience like here in this area, we actually have like a negative relationship between photosynthetic quantum use efficiency and fluorescence quantum use efficiency. And then only this positive relationship we observe quite often, we can find like when there's already like an increase in the non-photochemical quenching. And then under very strong stress, which goes into the photo inhibition, we see actually that fluorescence and photosynthesis can have like a negative relationship again, where we then see an increase in the photosynthetic quantum yields. So the question is like, okay, can we observe this with measurements in the field? So what I did then in Antwerp, I joined an experiment where we created a very strong gradient in phosphorus availability for maize plants. And we added different kind of levels of phosphorus treatments, and then had two treatments where we also had like pasteurized soil, so no arbuscular mycorrhizal fungi. And this we observed then were like extremely stressed plants. In the end, they actually, like in the last 2 weeks, the plant which were at like very low values of phosphorus treatment and were pasteurized soil, they died in the end. So basically we created a very strong gradient in photosynthetic efficiency. At the same time, I measured over 2 campaigns. with a light core to get leaf photosynthetic values and quantum use efficiency of PS2. And I measured with a fluorowatt and ASD to get sun induced fluorescence at the leaf. And then we compared the leaf along this gradient, or like we compared always 10 leaves, and then compared the fluorescence yield and the quantum use efficiency of PS2 for the different treatments. And what we observed was actually this relationship we would expect, that under low and no P limitations, we see that we have here light limitations because we had to use artificial light, which only had around 400 micromole. So we have here in this area actually like a light limitations effects. Then with increasing stress, we see that we are like on the slope we usually observe within the field. And then only like under the extreme stress conditions where we had like pasteurized soil and very little phosphorus treatments, we see that the plants, when they were actually dying, that the fluorescence yield values were like increasing again. So, but these are measurements on the leaf. So how does it actually look at a canopy level? And here I would like to show like some results from Martini et al. From 2022, because it nicely demonstrates like one of the problem why it is so hard to observe this kind of behavior. So what you see here is from an experiment in Majadas, which is a savanna ecosystem. And what they show is actually like the photosynthetic efficiency values and the fluorescence efficiency values under increasing stress levels. This is here indicated with the size of the dots. And what we actually have to do with this plot is we have to invert it to compare it to the right plot, because now we are to have the fluorescence values on the epsilon axis and the photosynthetic values on the right axis. And what you can see is that we observe this similar relationship, but with the inverted relationship, so where the plants are under extreme stress, we have here a shift in where we actually locate the increase again. And this is like one of the difficulties and challenges if we compare over like a longer season, because one dot is basically 1 day in this plot. So if we have also like a strong seasonal trend, this shift or like this nice transition from the changing relationships, which we can see, for example, here, or which we can see here in the concept plot, we might not observe that directly because there's like a shift in these relationships. That's one of the reasons why it is so tricky to see these relationships like at a canopy scale from measurements in the field. Another reason is that we have structural effects, which makes it very difficult to interpret the signal, because when we have changes in the leaf angle, which is usually coming with recent stress level, then we have also changes in the reflectance and fluorescence values, and this is make everything a little bit more complicated. But this is something which, in this experiment, they actually excluded because it's a very stable leaf angle here in this experiment. So this is why this shows so nicely these relationships. So what we have to do, or if we want to have any chance to observe the nonlinearity driven by the physiology, we have to account for the variances in SIF, which can happen due to retrieval uncertainties, but also, as I already mentioned, changes in structure like leaf angle, but also changes in the pigments, for example, due to chlorophyll concentration, and to be able to link it really to the physiology. So, and these are the two big challenges. First, how can we interpret this nonlinear relationship? And second, how can we actually get to this nonlinear relationship because of the structural and pigment influences which are quite dominating the signal? And here I want to mention that especially like the scattering effects in the far red fluorescence, the reabsorption effects in the red SIF, and then, which is quite often neglected, but I think is very, very important, is like how do we properly estimate FAPAR? Because a lot of our normalizations depend on a good idea what FAPAR actually is. And I think we can here, or we have to maybe improve some of the current methods to get a better FAPAR. But today I would like to concentrate on the scattering and reabsorption effects of SIF. So what are they actually doing? So here you can see the scattering effect. And what we observe is that when we have unstressed conditions, there's a certain kind of photons which actually can arrive at the sensor, and the other fluorescence photons are basically scattered away. And then with a change in leaf angle, we basically receive a different kind of amounts of fluorescence values. This can be like more, it can be less. It really depends on the leaf angles and also like the structural effects we can observe. And to account for this, we need the escape probability of fluorescence. Here, the F escape 760 for the far red. Zheng proposed in 2019 a method to actually estimate F escape on remote sensing based approximations that he uses here, the near V, which is the near infrared reflectance of the vegetation, which is basically NDVI multiplied with the near infrared, and then divided by FFR. And from this, by then normalizing the fluorescence values by the F escape, we can calculate how much fluorescence is actually emitted by the photosystems. So that's the idea of this correction. For the red fluorescence, it's a different kind of challenge, because here we have very little scattering problems, but we have reabsorption, because the red light can still be reabsorbed, and what is quite often reabsorbed. So what we see is that within the leaf and then within the canopy, the red light is absorbed, so that not that much red light is actually be detected by our sensor. And then when we have like a decrease in chlorophyll content, we actually receive more fluorescence values. And this is what you can see in this nice graph, that with a change in chlorophyll, the interpretation of the first peak in fluorescence becomes quite tricky. So we need also the escape probability of the fluorescence. And for this, you et al. Made a suggestion to actually get the escape probability by normalizing the true reflectance of the red fluorescence, which is here approximated by the reflectance at 687 nanometers by the NDVI square, then divided by FAPAR, and then basically get in theory the escape probability, and then you can calculate the fluorescence in the red emitted by the photosystems. Okay, so much to the theory of how we can get rid of some of the structural problems. We set up then an experiment to compare leaf measurements with canopy measurements, and to see, okay, how do actually, when we measure at the same time the leaf and the canopy, how do red and far red fluorescence is related? And then we use the escape probability models to show or to see if the relationship are actually improving. For this, I set up an experiment in Antwerp, where we set up 10 mesocosms, 5 control and 5 drought. And for each mesocosm, we measured at the canopy with a flux system. And then from the flux, we have 2 spectrometers in the flux. So one spectrometer I use for the fluorescence, the QO Pro, and I use the flame to measure at the same time with a fluorwatt the leaf fluorescence values. And then I compared this to more or less at the same time measured values with each other to see how well they match. And we had 7 campaigns during 1 month, where we had different kinds of environmental conditions, and then also checked like, okay, how do the signals actually behave under increasing drought stress? But first, I compared the leaf and canopy measurements of far red fluorescence, and what we see that when we compare the uncorrected, the slope is not close to the 1-to-1 line, but this is, to be honest, also not realistic to expect, because we have 2 different measurement systems which work in a different way when it's about how much light is actually coming to the sensor. So the fluorwatt, we have to align to the sun, so we have different light conditions. But what I would expect is that they show an acceptable correlation between each other, so that they're not completely off. And what we see is that uncorrected, they already show a correlation of 0.34. But then when we correct with the escape probability proposed by Zheng, that we can improve on this quite a lot. So that was already like a nice result to see like, okay, we can compare the leaf and canopy measurements, even though the slope is off. Then we did the same for the red And we found, unfortunately, like a decrease in the relationship. So it became much more scattery and it didn't work. So this was very surprising because many other studies, we see that this correction works actually quite fine. So there was like this, okay, why doesn't it work in our case? Why do we actually lose the strength of the correlation? So to understand what is actually happening here, we run the scope model to simulate the escape probability for our measurements. And so we retrieved the escape probability from scope and then compared it with the escape probability calculated from Leo et al. And we see also here that scope and values from Leo did not match at all. So if we look into the formula by Leo, the idea is that we need the reflectance of the red of the vegetation. And they propose that when we measure the total red reflectance and multiply it with the square NDVI, that this is basically the red vegetation. So we tested this for our experiment and had in mind, okay, what makes our experiment now like so different from the other experiments where Leo was successfully used? And yeah, what is obvious? I mean, first, we have strong leaf angle movements within a potato plant. And second, we have like the soil influence because there's like no understory and we have directly the soil. So this might have like an impact. So what we checked is how the reflectance of the vegetation plus the soil is actually changing with leaf inclination angle. And this we did for vegetation plus soil and the reflectance only of the vegetation. And what we found is actually that in the red, so basically in this region, even though the overall contribution of the soil reflectance is lower than in the far red, we observed that with an increased leaf inclination angle, so the leaves are like going down, they're wilting and they're hanging down. And the more they hang down, the more contribution of the soil is actually like have an effect on the reflectance. If we compare then the red of the vegetation and the red total, so the total is basically what we would measure with a spectrometer, like on over the target, we see that with increasing leaf inclination angle, these two relationships are actually like diverging. So what we want to have is a vegetation index, which is sensitive to the leaf inclination angle and also can account for this diverging pattern. So to correct for basically that we receive more influence of the soil. If we check how the NDVI square is actually performing for this relationship of the red of the vegetation, the red total, we see that it is like a logarithmic relationship. So it does not do this job quite well. So when we then compare the red reflectance of the vegetation estimated from the NDVI and then compare it with red reflectance of the vegetation estimated by scope or simulated by the scope, we see that we actually get a negative correlation between these two. And only at very low leaf inclination angles we have a positive relationship. So at leaf angles which are like not changing a lot and they are like quite low, then the NDVI might still work. And also if there's like not a lot of soil contribution because we have an understory, then we would not have this problem that we have this non-functioning relationship. So what we then checked is if the near V can actually do the trick. And it's a little bit counterintuitive why using a near V, so near infrared reflectance signal for the correction of red fluorescence. But it actually accounts very well for this relationship between the red and the vegetation and the red total. And what we see that we have a much stronger linear relationship, which is also positive. So we simulated this along a leaf inclination gradient. This is the upper one. And then we compared the same simulations then with retrieved values from our measurements. And we see that we basically have the same patterns and that we also have a stronger gradient in the leaf inclination angles in our experiment, which explains why the NDVI did not work in our case. So we changed the NDVI square with the near V square in our simulations, and then see that scope estimated escape probability of the red and the one from near V is actually shows a much stronger relationship than the Leo model. So basically here, the message is if you do not have an understory and you have very little leaf inclination angles, the NDVI is fine. But if you have a strong soil contribution, you have no understory, and you expect a strong leaf inclination angle, how it is often with crops, then you might consider to switch to near V. We then also compared again the canopy and the leaf, and we see that also here we improve quite a lot the correlation between the red at the leaf and the canopy. So we use these two corrections, and now we want to see like, okay, how does this fluorescence values, and here especially the quantum use efficiency values, how do they actually change with increasing stress? As I mentioned before, we had like a drought treatment and a control treatment, but then also like changing environmental conditions. So what we had to do is to regroup the drought and control treatments to 3 stress groups. So in stress group 1, we put all the measurements which showed like low vapor pressure deficit values, so low atmospheric drought conditions, together with high relative extractable water values. So a relative extractable water of 1 means basically like the soil is saturated with water and water is completely available. Then a second group where we had still like relatively low vapor pressure deficit values, but already much more severe drought conditions. And then a third stress group with very high vapor pressure deficit values and drought conditions. So and then we compared these 3 stress groups along the quantum use efficiency of fluorescence in the red. And what we nicely see is that with increasing stress, we see a decrease in the values at the leaf and the canopy. So they both show the same behavior, and we found a quite significant relationship in this decrease. So this is also what we would expect, that with increasing stress, we see a decrease in the quantum use efficiencies of fluorescence. We did then the same for the far red values. And yeah, here I was a little bit puzzled, because even though we see that the values are decreasing, we see the same patterns, they are much less pronounced than in the red fluorescence. And at the same time, we see a strange behavior here for the unstressed conditions, where we actually have detect lower values than for the others. I try to understand what exactly is happening. So what we then compared is the coefficient of variation of all the measurements which are measured within one rotation of the pot. So we just compare, like we measure 16 times and then look at the variation within the 60 measurements. Then we turn the pot with the idea that the physiology should not change in this time, but the leaf inclination angle, so the position of the leaf to the position of the sun, is actually changing and get more variation in it. And then we compared, this is then basically the rotation group, where we then compared the coefficient of variation between this rotation states to see, okay, how does this affect when we compare different kind of leaf inclination angles with each other, and this then for the 2 signals. And what we found is that the coefficient of variation for the red is by far smaller for the red than for the far red. And then within the far red, we see much higher uncertainty range variance within the unstressed group. And I'm not 100% sure what could influence this. I expect, or I think it has something to do with how much variation we actually see within the leaf angles when they are not under stress conditions, that under increasing stress, that the leaf angles are more aligning to each other because they are stressed. But I'm not 100% sure. And this is something we still need to explore. Is this something which is really a thing? So do we really have a stronger impact of the leaf inclination angles in the far red fluorescence? Or is this something which is just for this experiment? You might ask, and of course we asked also ourselves, does the proposed adaptation of the F escape values of 687 are actually transferable to other targets or other experiments? So what we did, we then used a different kind of measurement data set to actually check, like, okay, can we also improve our estimations of fluorescence in the red by using the near V instead of the NDVI? What we now did in Leipzig is we set up a new experiment field within the Arboretum Großbösner, where we have an area. An arboretum is basically like a zoo for plants, where we have 100 different species. This 100 different tree species have 5 replica, which is randomly planted. And then we picked, unfortunately, we only have 2 flux sensors, so we can only measure in 2 trees, but we picked 2 of these trees to then see how actually like fluorescence is changing in time and if they're comparable or where we face problems. So we have 2 tower systems, and then with these tower systems, we measure at the same time with monopumps the electron transport rate. And then we have a system developed by Thea Kattenborn to also measure the leaf angle distribution or the average leaf angles over the day. And this is basically working with RGB cameras, which are making every 5 minutes an image, and then convoluted neural network machine learning algorithms, which are trained to estimate the leaf angle from these images. And then from this, we can derive the leaf angle. If you're interested in this method, I can recommend this paper. So the idea is that we get information about the fluorescence from NADI measurements. Then we get information about the physiology of the plants from the electron transport rate and the quantum use efficiency of PS2 from the micropumps. And then we get information about the leaf angles from this camera system. We checked now, how does the red fluorescence actually relate to the electron transport rate if you do not correct for the escape probability? And how does it improve if we correct with the NDVI approach from Leo? And we see that actually here it works. So we get an improvement in these relationships for the beech and the lime. So we here measure beech and lime. So in red, we have the Fagus sylvatica, and in blue, we have Tilia cordata. And yeah, basically, we then checked also how it works with the near V, because it's important that also our approach should be transferable to completely different canopies, and also how it behaves if there's like not a strong understory effects. And here we actually see that it's very close to the approach from the NDVI and actually even improves a little bit. But I would not overinterpret this. But important is that it is very close to the other one. And then we also wanted to see like, okay, how is actually red fluorescence comparing to far red fluorescence? And here we see something really interesting, because actually the red fluorescence after the correction worked better than the far red fluorescence after the correction. And here we are now like going into better understanding why actually the far red is tricky, especially for the Tilia cordata, for the lime tree. And one of the reasons is, or what I expect is actually the much stronger effect of leaf inclination angle changes, because if you know how Tilia cordata is actually changing the leaf angle with stress, it can be quite dramatic. And this is where I think that actually far red fluorescence is strongly influenced by the leaf inclination angle, which then changes the scattering effects, which is not fully compensated by just the near V. So this is something we are exploring at the moment, what actually is going on here. And with this, I would like to end with my take-home message that it is essential to accurately estimate the escape probability, because this is basically like super important if we want to interpret the dynamics in sun-induced fluorescence. And then the second one, that red fluorescence is a very valuable signal, and I think it is a little bit underused. The main reason there is especially like the reabsorption effect, but if we can get rid of the reabsorption effects, I think it's a very promising signal to interpret plant physiology and electron transport rate. So red fluorescence in particular important for proximal remote sensing. Of course, if we go to airborne or satellite, we face more challenges when it's about the uncertainty of the retrieval, which is quite severe in the red fluorescence. So the importance of red fluorescence, for especially in proximal remote sensing, I think we should explore this more.